In this video, I'm going to review some organelles and then discuss osmosis. So the first organelle I'm going to review is the nucleus. And this is mainly where your DNA is stored. Let's draw ourselves a picture of the nucleus. So here's its membrane, and notice it has a few holes in the membrane. So first thing we had is the nuclear membrane. And in this membrane we have the nuclear pores. And this is how things enter and exit the nucleus. We know that the nucleus contains DNA, but DNA is in this loose, all over the place configuration, and we call that chromatin. But it's still just DNA. So DNA in loose chromatin form. Lastly, we have this substructure, and this is the nucleolus. So last is the nucleolus. And here we have a substance called ribonucleic acid, which is abbreviated to RNA. And this helps create another organelle called ribosomes. Ribosomes are responsible for creating proteins, or we will say protein synthesis. Protein synthesis. Okay, so if you look here, we have a ribonucleic acid makes ribosomes. And once it's made, it exits the nucleus and goes on to the next organelle. So the ribosomes, they exit the nucleus and they reside in the rough endoplasmic reticulum. And this is abbreviated to RER. So let's draw that. So again, here's my nucleus with a few of the pores, and continuous with this membrane, we'll have that next organelle, the rough ER. So a few pores, and then like a flat outer covering. And it's called rough because under a strong microscope, you can see that it's studded with these ribosomes. So that's why they call it rough. Rough because studded with these ribosomes. One thing I forgot to mention in the nucleus, this is where something called transcription occurs. Transcription is part of protein synthesis. So on our way to make proteins, it reads DNA and we create an RNA template, and all this occurs in the nucleus. But we'll speak about this more in a sec. Okay, so here's the rough ER, rough again because it's studded with ribosomes. And now this is where, in the ribosomes, this is where the next stage of protein synthesis happens. We call this translation. Translation takes RNA and turns it into protein. So let's kind of go over an overview of protein synthesis. So our DNA starts here, becomes RNA, exits the nucleus, and then it fits through these ribosomes and out pops out our proteins. or the precursor to complete proteins. So let me review that. So in the nucleus, this is where DNA, an RNA template is made from DNA. And again, this process is called transcription. And then outside the nucleus, our RNA is turned into protein. And this occurs in the ribosomes of the rough ER. Ribosomes. And again, this process here, this is called translation. Okay, so now graphically, we know we have our double-stranded DNA structure composed of our nitrogenous bases. Now, an enzyme for RNA is going to read one of these strands, so we're going to have a single-stranded RNA template, kind of the instructions for our protein. Then it's going to enter this ribosome. You can think of this as a kind of like a factory that gets its instructions. So it's going to fit through this, and out is going to come out amino acids, which are the building blocks for a protein. So amino acid 1, then I'll abbreviate this to just AA, and then here would be AA3, and so forth. So this tripeptide, not a complete protein yet, but once you add many more amino acids, do some post-translational modification, then we can call this eventually a protein. But yes, that's pretty much it for the rough ER. The next organelle that is also adjacent is the smooth ER, and we call it smooth because it's not covered in ribosomes. And no ribosomes means this part doesn't undergo protein synthesis. So let's see what we got. Here's our nucleus with a few pores. And then we have our rough ER. And then we have the smooth ER. You can tell it's rough or not by the presence of these ribosomes. Okay, so we have the rough ER, and this would be the smooth ER. Okay, your smooth ER does a few things. One, it can store calcium. So to help you remember, think of muscle cells, which requires calcium for contraction. Think of muscle cells, they require calcium for contraction, among other things that a cell uses calcium for. Second thing is, it's responsible for detoxification. So this reminds me to think of the liver. Think of liver cells. Probably has a modified smooth ER. And we also have lipid production here. We mentioned a few lipids in this class. One are the triglycerides. Then we have steroids. If you modify steroids, we can make cholesterol. And then lastly, we have our phospholipids. And last thing for the smooth ER is the protein products that come from the nucleus, go through the ribosomes, they pass through the smooth ER, and to get to the next organelle, they're going to leave in vesicles. So these little containers, And these containers will take him to the next organelle, which is the Golgi apparatus. So to Golgi apparatus. Okay. So last is send products in vesicles to the Golgi apparatus. Okay, and this is our last organelle for the day. And you can think of the Golgi as kind of the UPS of the cell. Modifies things, and it also tells it where to go. So again, let me draw my smooth and rough ERs. Okay. So here's my smooth and my rough ER. Now for the Golgi, it kind of looks like a stack of pancakes, folded together. So there would be my Golgi apparatus. As these vesicles come from the smooth ER, it kind of merges together, and then via endocytosis releases its contents into the cell. So whatever contents were here will get released just like that via endocytosis. So comes here, say this is a blue product. This blue product will enter the Golgi apparatus. So just a quick recap, this area is the nucleus, this is the rough ER, this is the smooth ER, these guys are vesicles, and this is the Golgi apparatus. So your Golgi tags things to mark them for locations inside or outside the cells. So let's say it tags products. So they can go to the appropriate location. And it could be a location inside the cell, or it could be a location outside the cell. Depends on the product. And this process of adding things or taking away things, so one other thing it does, it can add attachments to products, or it could remove attachments, or we can remove pieces. So these two things, we call this post-translational modification. Post-translational modification. Because this process happens after translation. So DNA to RNA, this is called transcription. And then RNA to protein, this is called translation. So this changes by the Golgi happens after that. And then I will see you guys in the next video.